Hello, everyone. It's loud, isn't it? We have a biodiversity crisis, and sometimes at meetings like this, you wouldn't know it. It was actually the next speaker, Donna Agosti, who suggested we have this symposium because both infrastructures for biodiversity and the biodiversity crisis are two things that really concern people from Tadwig. It was really nice to see this, I think just last week or the week before, in one British newspaper, showing the Living Planet Index and getting the biodiversity crisis into focus to the general public. And all of the infrastructures we're going to hear today have a role in getting the message out there, getting it up there with climate change as one of the big problems we have to solve in the world. Anyway, I won't rabbit on about this. I will go straight on to introduce our first speaker, who's going to talk about the Biodiversity Literature Repository and Treatment Bank, Donna Agosti.
Thank you, Quentin, for this introduction and organizing a workshop on the biodiversity crisis. Thank you for giving me a chance to talk to you, such a fantastic crowd. 30 years ago, we had at that time, the biggest meeting in the world, the Rio Conference, which put the environmental crisis on a global stage. Among the three topics, there were the biodiversity crisis. And three of them, there were two things which were important for us. Like we need a general measure for conservation and sustainable use, and we have to monitor biodiversity change. And to monitor biodiversity means we have to figure out what's out there, how many species do we have, what do we know about them, and where do they live? Are they invasives or not? And all we know about them, at that time at least, was in our libraries, because we communicated building libraries. But we call them the print and PDF prison. So what do we actually want? We want to have publications, we want to make them, the data objects, citable, and we want them reusable. We want to make them reusable, not just to create a catalog of life that we know immediately, how many species do we have, what do we know about the species, but we want to also open up all this fantastic important information to conservation and to the genomics people and to the wider public. So we have to figure out ways that we open up our library. It's an impressive thing. So there are about 500 million pages printed. There are more than 1,000 journals publishing new data about annually, so it's a huge amount. We believe we have maybe 2 million species now described. We have 20 million, maybe 100 million taxonomic treatments. We have about 18,000 new species described every year. And in that, there are billions of facts. It's a huge amount of things, probably more than all the specimens in our collections. But only about 10 to 30% of the knowledge is digital. Most is unknown known knowledge. And the people are keen to get this, shows this. So in the 90s, we started to digitize all ant literature. And we had this cool idea that we PDF them all on the one hand, and we create a complete catalog of all the ant species and all the synonyms and everything. And each name is linked to a respective page in the publication. So we had two things. We have the full PDF, and each PDF had also its pages, and we could create very simple URIs. And you see the impact of that. Before, all the knowledge was centered at Harvard and a few natural history museums. And then we opened that up, and really people use it really, really big. The problem is that these PDFs, if there are human-centric. They're scanned. They're often scanned from xerocopies. They're made that for pleasing you with nice bitonal and fantastic colors and all of that. They're not machine actionable, so normally a machine cannot read PDFs. An overwhelming part of them today is in closed access journals, so we have no access anyway. There are no data in the articles which have links to other data. And working with this PDF doesn't scale up. That's one of the reasons we have no complete catalog of life, for example. So what we need to do, we have to automate as much as possible, automate, and only use people, the experts, when it's necessary. We have to make use of digital technologies, especially semantic annotations, and explain why. And make publications for begin with. That's a really important point, part of biodiversity knowledge graph. So don't publish PDFs, please, don't. So what do you want? We want to discover known biodiversity, so we have to make digital accessible knowledge. And of course, we can digitize everything, but we have to think what is what we want. So first of all, we have to get the content in a form that machines can read them. Then we have to keep like the minimal standards, like we need text flow. We have to know what's a figure, what's a figure citation, what's a caption, all these sort of things. And then we have to do some semantics to it. And because we want to know all about species, we have to have like a tag species. So we have to add tags around all the species. But we want to know more about a species, so we have to actually add tags to say, this part of the text, this is a treatment, and this is more detail. And we have to make sure we don't spend too much time in one thing, but we have to make sure that everybody and intelligent people can come in and continue working on that. And why annotations are important is shown here. So if you have a publication and you have a person name inside, it can mean anything, you don't know. But if you start add treatments, then you know this person has to do either is the author of a treatment, or is a collector, or is mentioned in that. And if you, for example, add a material citation, another more granular element, then we know, okay, this person has been a collector of this species, a specimen of this species in this publication. We only know this if you have these things. And luckily, this is all done for us. So we don't have to think about how we revolutionize our publications. Linnaeus did a really great job. He created not only the label, the Latin binomial, but he also started to create these treatments. So we have a treatment which is very well demarcated, the next one here, and so on. He had structure in it, nomenclature. He had a treatment citation, so already cited previous works. He had a distribution section, description, and he even had a section on biotic interactions. So it's very rich already. And the important thing is this is all based on specimens. So if you want to go, you go to the Linnaean collection and look at what Apis mellifera is, and we can actually find it. And now it's even a lecture tab, we have that. And we know it's part of the Systema Naturae. So today, we can make all these treatments fair digital objects. We make them accessible, in our case, in Zenodo. We created a taxonomic treatment object, which has all the elements which are needed to make fair data. And they are accessible for you. You can cite this. You can also get this in a machine-readable format, like JSON or so. And everything is, all the annotations are based on some standards we use, Darwin Core, TextPub, and others. And just a little bit thing, so when Rich talked this morning about the SEC part, so you see this species on the left-hand side, the original work, it's already implemented. So we can go say, okay, here is this Caledonia, in the sense of this author, 2019, who cites this original publication. And here you have the identifier for it, you can resolve it, and you get treatment. So essentially, what Rich talks about, there is an implementation. And if you actually look under the hood, this is very complicated. You cannot do too much, but it's, I talk too slow, no, too fast. But it's interesting because if you go into this, then you have more like material citations, which are even richer in that respect. So you can actually do much more. That means you can actually extract material citation, put them in GBIF, and all these points are now not just points or a holotype, but they're also linked to the original description. We can also take out all the figures. So you can actually look at figures, and each figure is linked again to a treatment, to taxonomic name, and the publication. And it's cool because you can actually reuse them. So you can use the figure which is from this publication, which is closed access, but you can reuse that in GBIF. Similarly, you can go in and take all the treatment citations out and build applications like Catalog of Life on the fly. But it's complicated, the treatment. So publication, there's much more to say. So in Bicycle, where we hear a lot about now, we actually go beyond that. So we want actually do a lot of linking beyond just the biodiversity domain and link to the omics, the taxonomic names and specimens. So we also, for that purpose, we write recommendations, which come out probably this week. We provide tools that you actually can link observation records and material citations. So you can actually link these two things. We have machines to actually do the work. So we have workflows. So we have a TreatmentBank, which is the production facility, and fair digital objects are stored in biodiversity literature repository. We work, so there's a lot of things processed. So there are more than 400 images, 700,000 treatments, and so on. And most of them are in GBIF and reused. We talk to publishers like Paintsoft or the CETA publishing group to make actually look in the future how we can do it. We have something really cool with that I'm going to close. Since August, we have now a fantastic workflow. So you publish, within 6 hours, your names are in a checklist bank. So you can look at this example, for example, here. This has been published, it went out, it's now in checklist bank waiting to be curated. Thank you.
Yeah, I think we have time for maybe 1-2 questions. Were there any questions online? You don't see any questions online either? Okay, because I know we're quite close on time, I will just continue on to the next one, and we can always discuss this later, and you can always find Donat as well. Joana, are you going to introduce the next one?
Hi, so our next speaker is Nicole Kearney, and she's going to talk about taxonomic literature and taxonomic databases, and I'll pass on to her.
They were downloaded. Okay. May I try? Oh, no, what's now? Yeah. Yeah. Right. Hello, everyone. I'm Nicole Kearney, and I manage the Australian branch of the Biodiversity Heritage Library. I also chair BHL's Global Persistent Identifier Working Group, which is now in its 3rd year of operation. Today, I'm going to talk about a project that Rod Page and I are working on to link the taxon names in our taxonomic databases to the source of truth behind them, the literature. I imagine that everyone in the audience is familiar with the concept of the biodiversity knowledge graph, which represents the links that should be present between our core biodiversity data sets online. But there's a missing link in our taxonomic databases. For the most part, we're still failing to link the taxon names through to the literature. And this is a serious impediment to taxonomic research and to biodiversity and conservation research, but it's also archaic and annoying. For example, of all of the name usages in the catalogue of life, 66% of them have a reference. There are 1.29 million references in the catalogue of life database, but 91% of those are flat text strings without a DOI or URL to link our users through to the literature. In fact, only 1.3% of all of the references in the catalogue of life have a DOI. And that's not because those those papers lack DOIs, we estimate that at least 25% of scientific species descriptions have a DOI. We're just not using them, and that's a problem. This is the profile page for the critically endangered Kuranda tree frog on the Atlas of Living Australia website. This species has an extremely limited distribution in far north Queensland. It was described for the first time in the literature in 2007. If you click on the Names tab on the Atlas of Living Australia website, you're taken through to the source of that name. The Atlas of Living Australia sources its faunal names from the Australian Faunal Directory. If you click on that accepted name link, you're taken through to the Australian Faunal Directory website. There is the very first description of that species in the literature. If I blow that up for you, you'll see that it lacks a DOI. This is that paper published in the Biological Journal of the Linnean Society in 2007. It most definitely has a DOI. That DOI is an essential component for this publication's bibliographic metadata. It should be included in every reference of that publication, whether that publication is in a scientific paper or in a taxonomic database. If I go back to that profile page on the Australian Faunal Directory website, you'll see that that citation is in the form of a link. It's encouraging. You would think that if you clicked on that link, it would take you through to the literature, but it doesn't. It takes you here to a page on the AFD or the Australian Faunal Directory website, specifically for this reference. That reference there is also in the form of a link, but it doesn't link out to the literature. It links back to this page. This is a dead end in our search for information. The same story applies to plants. This is one of Australia's most critically endangered orchid species. This is the name information for that species on the Australian Plant Name Index. Again, that link to the first description is in the form of a link, or the reference is in a form of a link. If you click on that, you're taken here. That page has two links. One is in the image of a book, but if you click on those, they don't take you through to the literature. They take you again to a page dedicated to that reference. Both the Australian Plant Name Index website and the Australian Faunal Directory think citations are important enough that every single citation in their databases has a dedicated page, but for the most part, they don't link out to the literature. So again, a dead end in our quest for knowledge. We need to do better than this because the literature is not only the foundation of our understanding of biodiversity, it also is the basis for our understanding of how that biodiversity is changing over time. If you cast your minds back to just before the global pandemic, one of the biggest global news stories was that Australia was on fire. Approximately 14.3 million hectares were burnt in those devastating bushfires, or approximately 35 million acres, which makes that one of the world's worst wildlife disasters in modern history. In 2020, BHL Australia received an urgent request from researchers studying the impact of those bushfires on Australia's native endangered orchid species. Those researchers wanted really easy access to the taxonomic literature, ideally from our taxonomic databases. The literature is not only the authoritative source of taxonomic knowledge, it also contains invaluable information about past distribution and abundance. To quote one of those orchid researchers, this is the information we need to refer to when we're doing a conservation assessment of a species towards having it listed as vulnerable, endangered, or critically endangered. It's therefore vital that we link out to these sources of information from our taxonomic databases to make it easy for these researchers, and ideally via DOIs. So to address this need, Rod Page and I contacted the Australian National Species List. We met with Anna Munro and Anne Fuchs, who's speaking directly after me in this session. They were really enthusiastic about this project and have been for about a decade. They sent us their citation data and we mapped that citation data to the DOIs for all of these publications. And those original citations that they'd sent us had been manually inputted into their database by a huge number of researchers, taxonomists. And so therefore those citations they varied enormously in their completeness and also in their format. And we were able to deliver them back clean, consistently formatted citations that we had pulled from that citation data in Crossref, the DOI registration agency for scholarly content. Now, this slide may not look very exciting, but this is the big reveal of my talk, because there on the Australian Plant Name Index website, thanks to Anna and Anne, live now is a DOI link. And if you click on that link in this taxonomic database, you're taken directly through to the publication. That's the first description of this critically endangered orchid species. And you can either download a PDF or you can click through to peruse this article on the BHL website. But why stop at Australian plants? We also sent a mapped citation database through, or a data set through to the Atlas of Living Australia. And just last week, Doug Palmer uploaded those map citations into the test website for the Atlas of Living Australia. Another reveal, if you click through on the names tab, there in the test backend of the Atlas of Living Australia is another DOI. If you click on that link, it takes you directly through to the very first description of the duck-billed platypus published in 1799. Rod Page has now uploaded his mapped citations for those data sets, the Australian Faunal Directory and the Australian Plant Name Index into Zenodo, where they're available for everyone, and he's now focusing on the IPNI citations. He's also uploaded those mapped data sets into TreatmentBank. This allows us to really easily search and query those data, that citation data set. There are 88,617 references in the Australian Faunal Directory dataset. If you search for DOIs amongst those, you'll see that Rod Page has mapped over 21,000 DOIs to those citations. That's about 25% of those citations that he's been able to find DOIs for, which is that figure that I mentioned earlier in the talk. Interestingly, or perhaps alarmingly, there are now more DOIs in this mapped data set on TreatmentBank from this small Australian taxonomic database than there are in all of the catalogue of life combined. That first reference there is for the Kuranda tree frog. There is still no DOI on the Australian Faunal Directory website for that species, but there is in TreatmentBank because Rod has done the mapping. So what we need now is for the Australian Faunal Directory to re-ingest that as they have in the Plant Names Index. So these DOI mappings are available to everyone, and the aim of this project is that TreatmentBank will enable us to link together all the occurrences of the same name in the literature, ideally via our shared identifiers. Just quickly want to address the 75% of taxonomic descriptions that lack DOIs and therefore sit outside that linked network of scholarly research. That number is decreasing thanks to the tireless efforts of BHL's Persistent Identifier Working Group, who are retrospectively assigning DOIs to the 60 million pages of our legacy literature on BHL. You can only assign a DOI to something if it's online. So over the last 2 years, my BHL Australia team has been madly digitising ORCID literature and uploading it online. So this is a shout out to my BHL Australia team and to the BHL community globally for making the world's biodiversity literature available online. So it's thanks to that foundation work and to the work that Rod Page has been doing for decades, creating mapping between names and literature, that he's been able to upload that mapping onto Zenodo and TreatmentBank and making it available to everyone. And he's now just last week reached out to IPNI and GBIF about getting these mapped citations into their databases. What I'd really like to see is this. I can retire once this happens, this is not live or available yet, but we're hoping that it will be. So linking from GBIF out to the citations. And I'd like to acknowledge this recent paper that was published last week, of which Donna is an author on. This is guidelines for best practice and for the citation of authorities of scientific names in taxonomy, encouraging all authors, publishers and editors to give the bibliographic source of the authorities of taxonomic names. I would argue that this applies equally to those contributing to taxonomic databases. So for all those involved in scientific publishing and all those contributing to taxonomic databases, please provide the bibliographic source of the authorities of taxonomic names, cite each description properly as a bibliographic reference, link each scientific name to its original description, and for goodness sake, provide the DOI wherever DOI exists. Finally, a quote from my co-author and close colleague, Rod Page: "Without links, taxonomic databases are little more than online collections of index cards," which is technology that was invented by Linnaeus in the 1700s. Surely we have moved beyond this. Thank you.
Thank you, Nicole, for the very nice talk. So is there any questions from the audience here for Nicole? Okay, so I have a question here from Quentin. You want to put it? Sure.
It was a very practical question, actually. That 75% that don't have a DOI, was that from an Australian checklist? Was that every taxonomic name of all taxa, or?
It's an estimate that Rob Page gave based on his entire kind of collection that he has of mapped data sets. It certainly works for the Australian fauna. That was, we actually got the same 25% when he investigated that data and mapped DOIs, but that was the estimate he gave me. And I see him as kind of the expert on all of this, seeing he spent the last couple of decades doing this mapping work.
It's a useful number to know and to track.
Yes, we're not 100% like in the same way that we actually don't know how many taxonomic articles there are on BHL or in the world. Like, it's a hard estimate to make. Not even all the taxonomy literature that's published now is given DOIs, unfortunately.
Thank you. There's another question here from the Zoom chat, which is from Sjovan Leachman. And the question is, anyone other than BHL doing legacy DOIs?
There are a lot of, so thanks, Siobhan. Siobhan is one of my close friends and colleagues. There are a lot of journals that are doing that for their own publications. So a lot of journals will upload their current or publish their current literature, and then they will do their own back issues of DOIs. So we often work very closely with those through BHL. But yeah, most people are doing it just for their own literature, their own publications. But a lot of through the BHL RetroPIDs project, the retrospective assigning of DOIs, we're working very closely with publishers around the world. So we're hoping that Natural History Museum in Paris, who spent most of the last 2 years uploading and assigning defining all the articles in their back issues of their journals, that they will be the next large organization to assign DOIs to their own back issues. So we're supporting all the organizations that are doing that too.
Thank you. Is there any other question?
Thanks. Great talk, Nicole. So there's a lot of taxon-based communities with vast repositories of literature, PDFs, names, and in many cases, actually like my own, the links between those names and the PDFs. But there's still nowhere really that we as taxon communities can sort of link all that up or put that into kind of like a more sustainable infrastructure. Right now, that sits basically on our databases within those little communities. So I guess maybe it's part plea, but how do we bridge that kind of taxon community gap to get that into kind of more of the public domain and outside of those little taxon communities like mine?
So you're talking specifically about the PDFs themselves or the mapping?
Well, I mean, a good example that I've got, so in terms of parasitic lice, 70,000 taxa linked to their PDFs. All of those names are linked. Most of those PDFs won't have a DOI, but we've got all that linkage basically in what is effectively a community database. But I've got no way of kind of pushing those PDFs to make them public other than in my own little space, and no way of kind of publicly sharing that without a really big amount of work, which to be frank, I don't have time for. So how do we get those communities to then contribute in that way to these kind of efforts?
I think there are a whole lot of kind of pulling together of all these things. And yes, you're right, it's a massive piece of work. Certainly the work that Rod's doing to work with a lot of these databases and pull them in kind of separately. I mean, we've done that now with the Australian Faunal Directory and the Australian Plant Name Index. And he's now, there are lots and lots of databases like yours that have that mapping. So pulling them together would be excellent. And yeah, maybe it's a similar sort of process that we can do that we've done with those two databases in Australia that we're hoping that using Checklist Bank and using Zenodo, we'll upload those and people can then access them. The kind of going backwards and forwards between the two is a lot of work. But yes, get in touch. We're out of time.
Just a quick question.
Essentially, it's not a question, it's a comment. So you can upload all this to the biodiversity literature repository and we can help you to do it. So ideally, it allows you to do more because of this copyright and all these issues. So that's complementary to BHL. But we are interested in work with these communities to not only upload them, but eventually also to extract all the treatments and make them accessible.
Do you want me to answer that?
Just a quick comment. Please do go and look on Slack. Rod has actually put some comments in there, and the answer to Vince's question is Wikidata, Wikidata, Wikidata.
Yes, and I think I should have emphasized that as well. Huge fans of Wikidata, and Rod is obviously the technical person behind all of this work. So technical questions to him, please.
So the next person we have up is Anne Fuchs online. Is she doing it live online?
Good morning.
Video, okay. Oh yeah, and a lot of links from Nicole's talk are online, apparently, on Slack. Sorry, do I need to kick off the... Ready, yeah.
Morning or good evening, depending on where you are in the world. I'm Anne Fuchs and I'm the project manager for the Australian National Species List infrastructure. And I'd like to present some recent work we have undertaken in this project. Before we begin, I'd like to acknowledge the traditional custodians of the land on which we meet and pay my respects to their elders past and present. The work undertaken in respect to this talk was initiated by a Australian federal government project to improve environmental assessment management. Australia is a large country with a diverse and unique biota, and biodiversity conservation is managed independently by each state or territory. States are responsible for collecting proponent data and state atlases, while the federal government manages environmental assessment and approval processes under the Environment Protection and Biodiversity Conservation Act, or EPBC. The overall project is being managed by the Digital Environment Assessments Program, or DEEP, and one objective of that project is the integration of systems and data across jurisdictions. To support environmental assessments, ecological and occurrence data is being held, which will be managed in a new biodiversity data repository, or BDR. Project designs decisions that affected the NSL or were of interest to the NSL were the agreement to use the Australian National Species List as the taxonomic backbone for the BDR and the use of graph technologies to deliver the repository. This solution built upon the long-term work of the Terrestrial Ecosystems Research Network, or TERN, and their use of linked data and the semantic web to deliver data and analytics for ecosystem research. TERN was also engaged to deliver a new standard for the exchange of ecological data for the BDR, known as the Australian Biodiversity Information Standard. The BDR project has two parts. The first part, ingestion of occurrence data from a range of sources, including point, plot, and transect biodiversity survey data with their associated data quality checks. And secondly, storage of this data in a graph and the publication to support open data access to all systems and users of the data. The BDR graph will use the Australian National Species List data to provide the taxonomic backbone. This solution enables up-to-date taxonomic changes to be readily implemented. So why was the Australian National Species List used for this work? It is a collaborative effort by the taxonomic community in Australia covering native and naturalised taxa with the objective of maintaining regionally accepted taxonomic names and concepts. The datasets in the National Species List have existing governance and support mechanisms to maintain this data over time. Currently, the Australian National Species List contains datasets with a name index and accepted taxonomies for each of the following domains. The National Species List is built on a relational database containing taxonomic, nomenclatural, and profile data. The infrastructure provides identifiers for and linkages between objects for names, taxon name usages, and taxonomic classifications over time. Additional information about the identifiers available in the infrastructure can be provided if required. As part of the project, the NSL was required to provide endpoints to firstly check the validity of the names being provided by proponents as part of the ingestion process, and to deliver a SCOS representation of the current taxonomy. The work undertaken for these services needed to meet the tight timeframes of the BDR project, and so they have been developed to meet the mandatory deliverables required. Where possible, the long-term vision for the NSL infrastructure was also incorporated, but by necessity, there's been trade-offs. The two services are based on scientific name identifiers. The incoming data provides scientific names and is mapped to a scientific name identifier. To then provide a mapping of this name to the currently accepted taxa, the SCOS graph includes the relationships between scientific name IDs. The first endpoint delivers a name check and returns the set of potential name identifiers for a requested name and returns the result in JSON. The check can be requested against all National Species List datasets or a specific one. The endpoint delivers a check of the incoming name. It's not a search. This will be provided as a separate endpoint in the near future. The processing will try to match the supplied name as provided, and if it fails to find a match, it will use GNParser to separate the name into components and retry the check. The reason for the two-step process is to try and handle differences in recording of data such as rank values and authorship and improve the match rate. The JSON output returned for each name includes the verbatim search term and if changes are made to the provided input, the process search string. The dataset searched, where NSL is all datasets, and the BDR required the provenance of the check algorithm to be transparent, so the GitHub release of the source code is returned in each record. For each requested name, a result set is returned. The match type indicates whether the name is matched fully, including authorship, or whether it matched to a name without authorship. This provides data quality checks when partial names are provided. The matched name details, including names, identifiers, and the nomenclatural status, and the taxonomic status of the name in respect to the classification is returned. Names include in the name indexes, but not yet included in the taxonomy, are indicated as unplaced. The delivery of the higher classification and distribution data were requested by the BDR to support additional verification of the results and are generated from the classification. The second service provides the Australian National Species List classifications as a SCOS output using JSON LD. It includes for each dataset the accepted names and their synonymy, excluded names, and names yet to be placed in the classification, for example, recently published names. The SCOS output includes for each dataset the tree or classification, including its current and previous versions. For the current version, its creator, publisher, and other attributes. All scientific names contained within the tree are described using a SCOS concept of taxon name. The taxonomic status indicates its status in the classification, and the hierarchy of taxa in the classification are described via broader than relationships. This is an example for Cycas basaltica. It has an identifier and its preferred label is its scientific name, and it is currently accepted in the Australian Plant Census. Cycas basaltica is part of a broader concept of cycas, and it has a homotypic synonym identified by another ID representing Cycas media var basaltica. To further describe related names within the SCOS, subproperties of SCOS XL alt label and SCOS alt label have been used to categorize the relationships, and these are currently defined using a local vocabulary. The SCOS output is delivered as a series of zip files generated nightly. A request with no parameters will deliver the most current file, and a list of available zip outputs are at that URL. Future developments include a consolidated Australian National Species List taxonomy with a single classification combining all the existing data sets and providing a single entry point, making delivery of data to clients who want a consolidated list such as the Atlas of Living Australia, the BDR and State Biodiversity Atlases easier. We will be extending the NSL graph to a full RDF implementation and developing new endpoints. We will transfer our existing services to a set which align more closely to the expected standards for the delivery of taxonomic data and adopt TDWG standards, particularly TCS2 when it is ratified. We will provide a consolidated website to allow queries and display across all Australian National Species List data and make statistics available, including things like the number of taxa and number of new publications. The new website will include governance details of the data. We're also involved in preliminary discussions with consumers about the need for a stable taxon identifier and we are participating in community discussions, including some of which are being presented at this conference. I'd just like to thank all of the people involved in this project and pass it over for questions. Thank you.
Good. Well, we're lucky to have Anna online. I think I see her there at the top. Can you hear us, Anna?
Yes, I can hear you.
Okay. Do we have any questions for Anna? She stayed up especially late to be here for your questions. So.
So this is a question from Claude Nozores to Anne. Does the backbone and repository also cover the marine domain? Was this a challenge, for example, limiting what species occur in Australian waters?
The data set that contributes the faunal data is the Australian Faunal Directory, and it includes Australian offshore taxa to, I think, the Australian marine boundary. So if the data is available in AFD, then it will be included in these outputs, yes.
Any other questions? I did have one myself, actually, very selfishly. I wondered about this scientific name authorship, and I'm particularly interested in people's names and their links to identifiers for people. And I noticed that the Darwin Court doesn't actually have a scientific name authorship ID as a term, although you could potentially use IDs in SCOS. Do you have any plans to link the authors to identifiers for them?
Yes, we would like to. I've started like preliminary looking at other sources for identifiers such as IPNI, and we would hope to have an exercise to actually try and allocate ORC IDs or Wikidata IDs where possible to authors.
Great. Any final questions? Otherwise, we'll let Anna, Woody wants to go to bed. Okay, we'll move on to the next.
Thank you.
Okay, so next we have Joe Miller that will talk about the new framework for GBIF, and I'll pass it on.
Great, thank you. Yeah, thank you, Quentin and Donut, for thinking of this session. It's nice to talk about a different topic than I usually talk about. And at the Secretariat and in the broader nodes community of GBIF, we've been talking strategic planning for the last 2 years, and it's coming to an end, starting with a new strategic framework. And I would like to go through that today. So I'm going to be, so people in the room that are part of our formal nodes network have been part of this process, but I realize that a lot of you in the room are not node managers, but of course I consider, and hopefully you consider yourselves part of the GBIF network, and GBIF and TAGWIG are one big family. So some of this may be new to you, so I hope it's of interest as well, because when I say the word we, I don't hope during this talk, which I will a lot, I just don't mean the GBIF Secretariat. Hopefully it means everybody in this room. So we're just ending our third strategic framework at GBIF. It was supposed to end at the end of 2021, but we all know what happened in 2020. We were beginning our planning, and our executive committee said, well, it's too complicated now with the pandemic. So we extended our strategic framework until the end of this year. So for the past 2 years, we've been developing our new strategic framework. Last year, our governing board agreed to our strategic framework for the 5 years ahead. And this year, the implementation plan on how we're going to achieve our goals in the plan. So our strategic framework, which has not really come out public yet because it starts in 2023, and I think we'll roll it out later this year in kind of conjunction with the COP biodiversity meetings in December. But we do, it's only a 3-page document. It's short and sweet, but we do mention the challenges of biodiversity loss and climate change. And we also talk of the urgent need for solutions to multiple global crises. So it's not just a document about infrastructure and people, but we tried to write into it a sense of urgency that we all feel about biodiversity and how we can, as a community and GBIF, make GBIF relevant in coming up with solutions. because we do recognize that biodiversity data is more complicated than just the occurrence of species in time and space. Organisms interact, they co-occur, they move, and they evolve, they do lots of things, and we're trying to capture that now. So in our strategic framework, we have a vision statement. GBIF's vision is a world in which the best possible biodiversity data underpins research, policy, and decisions. And I think this reemphasizes kind of the lane that we try to be in GBIF. We want to be the enabling infrastructure for data. You know, it's not our role to make the decisions in research and policy, but we want to get the best data possible in formats possible so that people can use it for those reasons. We have a mission statement, which is a bit more detailed, to mobilize the data, skills, and technologies needed to make comprehensive biodiversity information freely available for science and decisions addressing biodiversity loss and sustainable development. So this is, our mission is to get that data, put it in the format so that people will use it and make people aware of them so they can use it. So during this process, it was interesting. I'm not sure who came up with the idea, but I think it was within the Secretariat to come up with some core GBIF values. And we didn't have any of these in our previous strategic plans. And this actually got the most interest in the nodes community when we had our outreach events with the nodes community. And these are the ones we came down upon. So trust and transparency, collaboration and collective benefit, diversity and exclusiveness, innovation and integrity. And all these terms are meant just not for an infrastructure, but for the people network as well. We want trust and transparency in the data, but also in the people that are preparing the data. And I hope all of these you agree that these are part of your own work as well. So we went down with 4 strategic priority areas. If you're familiar with our former strategic plan, we had 5. And these have a little bit longer titles, but basically the first one is science, building evidence for advanced scientific research and understanding global biodiversity. We want GBIF to be relevant for science. Number 2 is supporting research or specifically policy responses and knowledge transfer to address these urgent societal challenges, which is what we're talking about now. So that's more on the science policy. So our short term for this is policy, science and policy. And the next 2 will be very familiar, enabling the network to meet future needs and challenges. So this is our people, our network priority area. And 4th, of course, is driving innovation to advance biodiversity knowledge. So this is the infrastructure and the data that comes from the community. So it's science, policy, network, and infrastructure and data. Okay, so these are all nice words and we wrote a nice document about them, but what are we actually really going to do? So how are we going to implement to reach these goals? So I'm just going to highlight 4 kind of areas that we are working on that I think will shift the way we are interacting with the community, and hopefully we'll be providing more useful data to address these global problems that we have. The first, thematic streams. Second, accelerating our interactions with the policy. That's really along with our priority area 2, distributed workforce, and a little bit on the alliance. So a few years ago, hopefully most of you have seen this paper by Heberlein et al. In 2021. Since we have been tracking the usage of GBIF data in the literature, did analysis looking at how users are using the data and really starting to take this information of how the uses are, how data is being used, and see if we can using that to inform our strategic planning. And we can see that there's certain areas that are increasing in relative. All these topics are increasing in gross numbers. You know, there's certain topics that are growing, certain topics that are lessening. So we took this information and we kind of built it into our work of trying to bring new communities into GBIF. So, you know, the core of GBIF has been the collections community. You know, we have the nodes, the ministries and museums. What we're trying to do in this thematic area is really trying to bring new communities such as evolutionary biologists or ecologists. That's on the left here. We have these communities who go to conferences, they go to their own conferences, they have their own journals, and they work in this milieu by themselves. And then the other side is what we're trying to do as a community here, data growth and data use. And we're see if we can build bridges between these communities. So over the past few years, and we've have new tools that we're using to interact with these communities, things such as data papers. We've had some expert groups to ask a community of potential users, what data do you have and how can you be part of the solutions? We've had thematic desks, help desks. We've had some data grants. We've had volunteers, and of course, these landscape reviewing papers. So we have these new tools that we're trying to make a bridge between these communities that are not submitting and using GBIF data to get in and part of the process. So that's part of what's driving the thematic roads. Next is a couple of examples. Of course, we have a program we call GBIF Health now. A few years ago, we did have I think this is only 2 or 3 years ago, we started an expert group in health on vector-borne diseases and included people from the WHO. They did a landscaping review paper on the use of GBIF-mediated data in this type of literature. That's submitted and now in for review. This expert group is being restarted. We did have a thematic help desk to help new data providers with specific questions of this type of data. And now we have a second call for papers in this. So we know there are communities out there that have relevant data in this place, but we're trying to give them the tools to come in. And we have this health communications as well. Similarly with freshwater, a very active research expert group, some data papers, landscape reviewing, and try to develop new partnerships such as the Ramsar. So this is an example of some of our data thematic work in conservation, food security, climate change, and health. So we're trying to bridge from data source to data use. We're also accelerating our interactions with policy. This is a diagram on the upper left from BERTnet. They did an analysis of the biodiversity needs in the post-2020 framework. What are the current uses and what are the possibilities for GBIF-mediated data in this framework? And they gave us some recommendations, and we are in the process of implementing them, especially in regard to the Montreal COP coming up in December. And we're very heavily involved with the CBD and IPBES. My colleague Tim Hirsch is really leading that effort for the Secretariat. In one of our tasks this year, we're trying to become embedded in the GEF, the Global Environment Facility, as a data repository for them, and started to reach out with other conventions. We also have a more distributed workforce now, not just at the Secretariat. We have research support, regional support teams in Africa, Asia, and Latin America, providing local research support in local languages. And these things is something that we can help also stimulate and expedite the data growth. And also distributive collaborative infrastructure. And this is probably a poor list because there should be more on here probably, but we heard about TreatmentBank. It's one good example, and it's providing new ideas for the community. But there's a lot of joint development with ALA, IDIG-Bio, DISCO, DataSite, and really trying to leverage each other's capacity. And finally, we're also doing the next step on the Alliance for Biodiversity Knowledge. So right now we have a call for it's a consultation out that is assessing what we have done in the past few years on the Alliance and coming up with some recommendations for what the Alliance can do and what an Alliance project office could do and a governing mechanism for that. So this is an ongoing work. We should have some final results in January, but some of their preliminary recommendations are that the Alliance for Biodiversity Knowledge should be in organizations and not people. Really should balance membership between data and technology and policy actions, because we know this is important to the community. Something I really want, clarity and transparency on the role of GBIF. And the discussions they've been having with the community really focus around 3 major topics: what the organization of the Alliance will be, the actions it take, and how collaborations will take place. So the next step is happening right now, and we'll have their report early next year that we'll bring to you, the community. And then kind of underpinning, hopefully you hear a lot about this week, is the GBIF, the Unified Data Model, which is our way of advancing as well. Thank you very much.
Thank you, Joe, for the talk. Thank you very much. Is there any questions from the audience? We don't have any questions online, but Quentin has a question.
I always have questions.
Good.
So indicators are a big thing,
Yeah.
and monitoring. And I wondered how much you expect scientists using GBIF data to be the creators of indicators, or whether GBIF will start creating indicators, or is it a bit of both?
So I see our role as enabling. So that's the new term that Tim Robertson is using, and I'd like to use it too, enabling infrastructure. So I think our role is to, if there is a data format that a group that is developing indicators would like, that would help them do their work, we could develop export formats of GBIF mediated data in a format that would facilitate that process for them, make it faster and easier for them, perhaps clean in a certain way. That's kind of our role. We don't have any experts on staff to make indicators, and we're not going to hire indicator developers, but we certainly have the expertise to get that data in the format right for them to be used in whatever way possible. So I think that's a very exciting a next step for us. And there's lots of interest in the communities that are building the EVVs and indicators for us to be in that role.
Thank you, Joe. Any other question? Okay. No. That's all. Passing on.
Okay, so next up is actually Joana Pauperio. Is that right? I will. Go ahead. So Joana comes from EMBL, the European Molecular Biology Laboratory in the UK, and is part of the Bicycle project, which I probably should have said is the project that's kind of the overall umbrella for this symposium. Okay, we're nearly there. You missed.
Okay, sorry for this technical thing. Today, can you? Okay, yeah. Thank you. So today I'm here to talk a little bit about ENA's and ELIXIR's perspective on connecting molecular data and the biodiversity research community. So we all know that molecular data is key to discover new species, to document diversity, to understand evolution and diversity, and for monitoring. There are several recent papers documenting the importance of genomic variation for biodiversity and monitoring, for detecting hidden diversity, and the importance of digital sequence information for biodiversity conservation. And we all know this biodiversity knowledge graph by Roderic Page, where we can see all the different areas within the biodiversity research cycle properly linked, but we still need to work towards this linking and towards improving the connection between molecular data and all the other aspects, so we can have findable, accessible, interoperable, and reusable knowledge and molecular data. So I'm sure some of you already saw a similar slide, and you probably all know what the European Nuclear Archive is. So we're an open platform for the management, sharing, integration, and dissemination of sequence data. We are the European node of the International Nucleotide Sequence Database Collaboration, the INSTC. And we hold data for over 1.5 million species. And we have comprehensive databasing infrastructure for archiving petabytes of sequence data and several tools and services for the management of the data. ELIXIR, so the European Life Science Infrastructure, has a recent emergent biodiversity community. So this community was initially established in 2019, but has an ELIXIR focus group. So a group of people that are trying to address emergence areas of interest and identifying a strategy that ELIXIR can follow. So they've been active for over 2 years with 8 of 9 modes with regular involvement. And in March 2020, they established an initial catalog of ELIXIR services support for biodiversity research. And in October last year, so a year ago, they started a journey towards becoming a community. They expect by 2023 to be an ELIXIR community. There is a group of experts that are working together towards driving development of standards, services, and training in the biodiversity research area. So these emergence community goals are to understand and catalog capabilities, interests, and ongoing projects that there is in the area across the several ELIXIR nodes, to develop connections with key external partners like GBIF, DISCO, LifeWatch, Bioscan Europe, for example, to recommend short-term activities that will advance the field within ELIXIR, to develop a longer-term strategy on how biodiversity can be supported by ELIXIR, and to coordinate aspects of potential grant applications. So one of the largest efforts that the focus group has been doing has been discussing recommendations for connecting molecular data and biodiversity. This paper was published in the end of last year, and it addresses 4 use cases that are good examples on how molecular data can inform biodiversity understanding. And then it has 7 general recommendations, which we believe are essential for the wider field of biodiversity. So these recommendations are here. So the first one is that biodiversity-related and molecular-focused infrastructures need to collaborate, and I think we're doing that or starting it. Taxonomies should be aligned and harmonized across domains. Metadata needs a better standardization and to be universally adopted. Approaches for managing molecular data need to scale up. Bioinformatics tools and services need to be prioritized. Training needs to be widely available to the community and sustained. And the biodiversity community needs to proactively seek common solutions that enable molecular technologies to advance biodiversity research. So both ELIXIR and ENA have been involved in the last years in several biodiversity projects. These are some of the projects that we are involved in, and I'm going to speak a little bit more about some of these. So these projects here are all biodiversity genomic projects that include our Tree of Life, aquatic symbiosis, and more recently the Biodiversity Genomics Europe, which joins together Bioscan Europe and ERGA, which is the European Reference Genome Atlas. So these projects are all large sequencing initiatives that are trying to scale up sequencing discovery, species interaction, and species dynamics. And they intend to accelerate the use of genomic science for biodiversity. And in these projects, we are contributing to the development of metadata management systems and interoperability resources. And we are trying to work towards a comprehensive, fair biogenomics data foundation. UNIUC is another project that we are involved. So UNIUC is a university taxonomic framework and integrated reference gene database. Okay, it's back. So UNIUC joins together the expertise of taxonomists and to curate available sequence data to have a reference database for eukaryotes. And there we are collaborating with them to display and connect these new developments and this reference, this taxonomic reference with NCBI taxonomy and with the records in INSTC. And you can see, you can't really see because it's a little bit blurry, but for example, from a data set they have curated, we now display the link between the sequence and the UNIUC taxonomy on UCMap, which is here, but I don't think you can actually see it because it's very small. Sorry about that. Another project that I'm sure you already heard about it and even saw a similar slide to this one, it's Bicycle, where we're working towards linking data between molecular data, natural history collection, taxonomy databases, and publishers. What we're working on at ENA is on data linkage and integration between molecular data and the other resources, data standards. We're also working on improving workflows for data submission and content enrichment, that there was a talk on these two aspects this morning, and also working towards building repositories for new data types when needed. One other aspect that is important to improve the standards of our metadata that is put together with the sequence data is the spatial temporal information. And there was a decision recent to make this information mandatory for all new data. This will be a future requirement, and it is expected to be implemented by the end of this year or beginning of next year. So in the future, we want to continue doing this work that we have been doing and also progressing on this towards supporting the growth of the network of networks for biodiversity, incorporating community feedback into the ELIXIR services development, and position ELIXIR as a go-to partner for European projects on biodiversity molecular initiatives that need data management expertise, and also biodiversity traits, taxonomy, literature efforts that need data integration expertise. We also want to continue to leverage ELIXIR platforms to support biodiversity research and continue the improvement of metadata standards and developing tools and workflows that support these enriched metadata that then we can link, and also continue supporting open data advocacy activities. So we believe that a strong foundation of interlinked biodiversity knowledge will contribute to increase our effectiveness in the response to biodiversity loss and ecosystem change. And I thank you all. And if you are interested in the ELIXIR biodiversity community, you can contact my colleagues over there on the slide. Thank you.
Thank you, Joanna.
Thank you.
I have a question online.
Okay.
From Carlos Martinez. Sequences for 1.5 million species, ? Are those some sort of molecularly defined units, most of them without their names, or formally published taxon concepts? Could you please clarify?
I think probably there's a bit of everything. I mean, we have a lot of information for formal species, but there's also information on, there's also molecular data that is published for things that haven't been described, for things that are considered unique, but haven't been described yet. So I guess we have a little bit of everything, but there is a lot of information for formally published species, yes. Because all of the sequences that are put into NSDC need to be linked with NCBI taxonomy. But whenever a species is not formally described yet, but there is information on it, it is possible to create a placeholder taxa for that. And there's also, this is more for genomes and assemblies, but there's also for general sequence data, you can submit things on the genus level, for example, or other ranks. Yeah.
Any questions on the floor? Another question online. Just think about your questions now. So from Ivan Lebara, questions. Speaking about metadata tasks, what are the plans from ELIXIR? On biodiversity, there is a major metadata standard EML, the ecological metadata language. Are you working on that?
Can you repeat that?
So, I think Ivan noticed you mentioned about metadata standards.
Yes.
And one of the major metadata standards in ecology is this ecological metadata language, EML.
Okay.
And the question is, are you working on it?
I can't really say specifically for ELIXIR, but I think if that is a standard that exists, I think everything is being taken into consideration and we're working with the community. So I believe we'll be working on that, but I can't really, yeah. Yeah, sorry.
He said plus 1,000 and I think
Sorry?
he said plus 1,000. So
Okay.
do we have anything else from the floor? Yeah, sure.
In linking the sequence data to the taxonomy, are you using the labeled sequence data or what they actually are? Because there are a lot of misidentified things in these databases.
So yeah, so I think there is an ongoing effort that will take a long time to fix. So we basically, the information there is what users provide, but now there are things that are being developed. For example, PlutoF, I'm not sure if you know, it's a platform from the University of Tartu. They do, and Link to Unite. So they do sequences assemblies for taxonomy inference. And for now, we developed a system that allows that information to be sent back to the public repositories through a clearinghouse. So the data can be curated. And then on our side, we need to contact the initial users and or put a curation on top of the records that will allow to correct some of the either misidentified or sequences that have been provided with no taxonomic information. And for example, UniUke is the same. So they're using public informations, they're running their phylogenies and their pipelines to be able to curate the taxonomy associated with those sequences. And that is being fed back to ENA and displayed together with records. So a record can have an NCBI taxonomy of uncultured fungi, but then it can have a UniUke taxonomy that actually has a species name associated with it. So that's a little bit one of the things that has been done within those projects. So I think there is a lot of things missing still. There will still be, there's a lot of work to do, but there are some initiatives already trying to correct that information.
More questions? Thank you. We'll continue on to the next talk by Chuck Cook. He's going to talk about sustainability, which is actually an extremely important subject for our domain. All of us can say projects that have not managed to sustain themselves.
All right. Yeah. Well, thank you. I'm going to take one step up here and talk about a kind of larger infrastructure project, which is all of biodata resources. And I'm going to just introduce the Global Biodata Coalition. Biodata resources are loosely speaking, any online resource which provides information of a biological nature. And we can talk a lot about how to define it, but certainly not in 10 minutes. But generally speaking, if you recognize it as a biodata resource, it probably is. It's a bit like what Richard said this morning about taxonomy, which is a taxon is defined by taxonomists. So biodata resources, well, there's a long history of sharing information in biology. Resources archive primary data and they provide added value information. This is what we've just heard about. There are thousands of resources. I'll talk a little bit more about that later. And they're all interconnected. And we've seen that in every single previous talk. All of the biodata resources are talking to each other, exchanging information. There's extensive collaboration and data exchange. And together it forms a rich and interdependent ecosystem, which we are calling an infrastructure and which is an infrastructure. And you can imagine, because they're all interconnected, that if there is a break in the system, if a major resource goes down because it loses its funding or something, that has a knock-on effect on all of the other resources. I've just said most of this. The biodata resources are a crucial infrastructure. They're used by all life scientists. It's a distributed infrastructure. They're all over the world. That's a strength because there's no one place that's crucial for the system, but it's also a weakness because it's very difficult to coordinate. We don't know who's funding them. We don't know where they are, but they all know where they're all interconnected. It's cost effective. Clearly, it's costs less to store research data and let other people use it than to regenerate it every time you need it. There's a huge opportunity in the community in sciences to improve this infrastructure to make it more useful for scientists, but it's under threat. That might be too strong a word, but as an infrastructure, and as with many infrastructures, the users of the infrastructure assume that it is well funded. You go out every day and drive your cars or get on the bus, and you assume that the local government that is making that road is maintaining it well. But occasionally you go to a place and the roads are really terrible. And then you start thinking about the infrastructure because it's not there. Well, the biodata infrastructure faces similar risks. It won't go away, but parts of it could have funding challenges. So the challenges are of two kinds. One, more science is being done, so there's a lot more demand on the infrastructure. There are new data types, and the existing resources are getting more and more data all the time. Joana didn't talk about the data flows into the European Nucleotide Archive, but they're increasing continuously, and in fact, recently exponentially. So on top of developing all of the new systems and working on all of the connections with the biodiversity infrastructure, the Nucleotide Archive also has to just deal with additional data flows in. Open data science policies increase demand, and there are sustainability challenges. So again, as I said, it's an infrastructure. We don't think about the funding too much, but in fact, the funding is a bit unstable and haphazard, short-term, and distributed unsystematically is actually a quote from Eric Green, who's the head of the NHGRI, which is part of NIH. So there's little coordination among funders of biodata resources. Many, certainly not all or most, even most perhaps, but many are funded by national and charitable research funders. And they have been concerned for a long time in trying to find ways to bring more stability to the funding for the infrastructure. And another challenge is, which I've just alluded to, is that the infrastructure is poorly described. We don't actually know how big it is or where things are, how many resources there are. So this need for a better understanding of the infrastructure and how to coordinate has been well understood for quite some time, particularly by the funders, because they're concerned with getting the most efficient use of the money they provide. So the funders, well, there's this publication from 2015, but this isn't, that wasn't really the beginning of the story. That's just the first public publication. But there was a concerted effort among funders and stakeholders, which is scientists and managers of some of the major databases and biodata resources, to try to find a way to bring some more cohesiveness to the infrastructure. And that resulted in the formation of the Global Biodata Coalition. And on the right-hand side here, there's some dates, but basically there were a series of meetings. NIH, NSF, Wellcome, and A-STAR in Singapore provided a little bit of seed funding, and we developed the Global Biodata Coalition from that seed funding. The mission, there are just two missions. One is to be a forum for funders of biodata resources to coordinate their efforts. So I need to emphasize that the GBC works for the funders. The funders give us money to do stuff for them. We're not providing any data, any money for databases or anything. It's a very tiny amount of money. It supports about 4 FTEs. And then the other major thing is to find ways to sustain the infrastructure. We're working on that. I'm not really talking about that today, but it'll be the subject of future talks and papers. And we're also focusing on identifying which of a smaller number of biodata resources are core to the infrastructure, most central or most important in a definable way. Just a quick slide on the left-hand side. Those icons. Those logos are the funders who are actually giving us money, and I have to say that it's a tiny amount of money. It just supports a few salaries and a bit of travel, like me coming to Bulgaria from the United Kingdom on Ryanair. And we have some observers over here, which are other funders who we hope will join. And the Secretariat at the moment only has 6 people, only 2 of them are full-time. And I just want to point out that our Executive Director, Guy Cochran, was supposed to give this talk, but was unable to come. But many of you know him, so I wanted to point out his name on here. GBC work program very quickly. I'm going to go very quickly because the last two slides are actually more important. But we have a focus on sustainability. This is working with funders. A focus on biodata resources, which I'll talk more about on the next slide, and a sort of paying attention to open biodata and global bioscience, which I won't talk about more. So sustainability is how does the funding infrastructure work? Working with the funders to find ways for them to share information so they provide funding in a more efficient way, and also trying to find ways for them to provide funding over a longer term. Most research funders, as you know, most research grants are say 3 or 5 years, and they tend to fund biodata resources and databases on the same funding cycle. But as infrastructure, you don't want to fund those on a 3 or 5 year cycle. Most resources should be there forever. You don't fund your roads over 3 years and say, well, in 3 years we'll decide if we still want roads, and then if we don't want them, we'll just let them fall apart. It doesn't work like that. But the funders are, because they're government funding and their budgets are a year at a time, they can't commit to 20 years of funding. So there's a fundamental conundrum here, and we hope to. I'm sure we won't solve it, but we hope to at least make some progress. Yes, and we have a couple of board working groups on open data strategies and sustainability, which we'll talk about at future meetings. Biodata resources, building community among users and managers. That's you people, most of you are part of the community. And we are also trying to characterize the infrastructure, which is at the bottom of this slide. So two of the things we're doing ongoing is developing an inventory of biodata resources. So we're working on that now. That should actually be finished by December and published early next year. The question is, how many biodata resources are there? Of course, that's an unanswerable question, but we do have a reproducible methodology, and we'll run it. We'll produce a number, and because it's reproducible and completely open, we'll be able to do it again in 4 years, and we can at least track changes in the infrastructure. And we're also running right now also a process to identify what we call global core biodata resources, which are biodata resources or databases that are crucial to the whole infrastructure. And if you want to understand what we mean by that, I can give you the links to look at the papers the paper we published on that. So the other thing, two things we wanted to mention are that the biodiversity infrastructure and data resources are well within the greater biodata infrastructure that GBC is concerned with. So it's within what we said on the slide, our sphere of interest. Life sciences needs information on more than just model organisms, and that sentence is in there because a lot of the data resources focus on model organisms, and they're very important. They're highly used, but they're not, of course, everything for all life scientists. So all of the life sciences is within scope, and everything is related. And as we said before, the resources don't exist in isolation. Everything there are data flows between everything. And standards, including biodiversity standards, are key to enabling those linkages to function properly. So this is also this slide is related to the COP15 process and the Nagoya Protocol, and ensuring fairness and equitability in usage and access to information. So the resources, the data, and it says biodiversity data here, but I think really all biodata is crucial to fairness and equity, both for scientists and for citizens. Open data is crucial for the same thing, for making sure everyone has access to information. And equitable benefit sharing mechanisms are also essential and should be built around an open data model and data standards. And in particular, crediting data sharing and credit, which is to say deposition of data into the resources, is crucially important. So there's a gap in the way scientists are evaluated. People are often look at publications, but how much data someone may have contributed to the infrastructure is not something that is considered as importantly. And one of the things GBC would like to work on is understanding or creating ways to develop ways to assess how people have contributed to the infrastructure. I think that was the end. So that was hopefully really quick, and really our purpose is to say Global Biodata Coalition is working with the funders to try to help make sure the infrastructure is around for a long time. And then I hope I've generated a few questions either here now or later at tea. So that's it.
Thank you, Chuck. So there's a question from the chat from Jorit. So what are you doing to track the overhead costs and design of informatics infrastructures and tools? And how are you reviewing the design, overhead, and technical debt in informatics infrastructures?
Well, that's a good question. It's a bit too fine for us. What we have looked at is trying to understand how much money is spent globally on supporting the infrastructure, on supporting biodata resources. And the answer to that question is that it's really a very difficult question. It's very difficult information to get, and we've kind of shelved it in the interest of doing something that is somewhat easier at the moment. What we would like to do is to understand, on the one hand, how much money is spent on research that generates data that goes into the infrastructure, and on the other hand, how much money is spent supporting the data resources that store and add value to the data generated by the research. So what we think is that the cost of supporting the biodata infrastructure is far less than 1% of the cost of research data, maybe even less than 0.5% or 0.2%. We don't know, but it's a very small number. But we don't actually know what that number is. It's very difficult to get a hold of. And if you want to send me an email, I can send you a long description of how difficult it is. But I will say that I talked to the OECD about doing this. And for those of you who don't know, the OECD spends a lot of money gathering statistics for all kinds of things like this. And they told me they couldn't do it. They tried and couldn't do it. So it's a tough thing to do.
Thank you. Is there any question from the audience?
Thanks a lot. Very interesting talk. I was wondering, so you're mostly focused on the hardware. So data needs to be stored on some kind of hard drives.
Really.
And
well, we're focused on the resources.
yeah,
And they're not really hardware. Most of the money goes into salaries, but yeah, there's some hardware aspect to it.
okay, but it's more the kind of resources that you can touch if you would go to a server room. I was wondering, what is your perspective or your connection to the soft resources that are necessary? Like, for example, organizations like TDWG who are developing the standards, who are actually allowing to connect these more hardware.
Yeah, that's it's like you basically, that's why I'm here, because this is our connection to standards. There's we're interested in standards, very important, crucial to the infrastructure, but our focus is on maintaining the databases at the moment. The other side to this is also software. So there's analysis tools and everything connected to the data resources, which many of you all of you are probably using, and many of you are generating. So those were also a little bit less connected to, although Chan Zuckerberg, which is one of our members, is also working on the software side of it. So So the answer is we know about those things. We try to talk to standards people and to software people as much as possible, but we don't have enough money to do everything.
Thank you. And thank you, Chuck. I think we're going to move on to the next talk, and then we can resume these discussions in the end. Our next talk is Colleen Royot, but she's not online, but I think Ivan is online. Yes, we'll be using the video.
Hi everyone, I'm Colleen Royot, a PhD student at the French Biodiversity Data Hub PNGB, and I'm going to present you the French EBV authorization pilot using Galaxy and ecological metadata language. I'm associated with Olivier Norvez, Marie-José, Elie Arnaud, Julia Sananicon, Sandrine Pawan, and Yvan Lebras from the PNGB as well. Jean-Baptiste from the French Natural History Museum and Sorbonne University, and Dominique Pelletier from IFREMER. So to operationalize EBVs, we started from the EBV workflow framework published by Kissling et al. In 2017. It presents a complete generalized workflow from raw data to EBV data products and indicators. So right after raw data and observations are EBV usable data sets that are pre-processed data, then EBV ready data sets that are formatted and harmonized data sets, and finally derived and modeled EBV data that are models produced from the data. Indicators can be inferred at each stage of this workflow with different description levels. So in addition to this framework, we decided to capitalize on the existent data analysis platform Galaxy. That is a web platform to share and process research data and permits to produce tools and scientific workflows that are accessible, reproducible, transparent, and can be peer-reviewed. It permits easy access to cloud analysis and high-performance computing through interfacing of many programming languages. So basically, Galaxy permits to get a complicated script such as an R or Python script through an interface, so no coding skills are necessary. In order to adapt a specific data analysis script that can process only one type of input file as it is common in the life science, into a Galaxy tool, we have to atomize the script into fundamental analytical bricks to be generalized, and that will form each tool of the overall analytical workflow that was made in the original script. Therefore, the tools can be shared on the Galaxy toolshed, that is such as an app store with, if you will, where anyone can use our tools with the aim data type in the primary workflow that was sought out, but also with other data types by using other tools from the toolshed and adapting the workflow to fit other proper data types. So these data types needs to be adapted to the analysis as well, of course. You can't analyze any data with any tool. So here are some of the EBV workflows already available on Galaxy and how it is reparted through the EBV classes. So we have the RATSEC workflow in the genetic composition. Then the stock and Pampa workflow are either in the species and the community composition level. Then the regional GAM workflow goes from species population analysis to species traits analysis. And finally, the OTU workflow goes for community composition. So I'm going to talk a bit more about the stock and the Pampa workflow that we implemented. So first, the stock workflow has been adapted from the usual analyses that are made by the French Common Birds Temporal Observatory, STOC. This workflow starts from occurrence data to preprocess and filter data to produce and analyze community metrics, such as the Community Specialization Index, CSI, and population abundance analysis as well. Then the Pampa workflow comes from the Pampa application that has been developed to permit Marine Protected Areas Manager to analyze data easily. Here it starts from raw abundance data that are preprocessed using a variety of existing Galaxy tools. So here are some examples, and here is the workflow we use to adapt another type of data to this workflow. So this workflow permits to analyze data at either community or population levels. And at each level, this workflow computes metrics such as Shannon index, for example. Then it produces a generalized linear model to evaluate the effects of site, year, and/or habitats on these metrics. And finally, it outputs time series plots of the computed models. So these two workflows have been implemented with many red flags and trainings to avoid any misusage and biased conclusion making.
Thank you very much, Colin. So it's Yvan Lebras now from PNDB, and I will make the transition towards the use of data and the focus on data related to this operationalization in EBV. So for this transition, I can start by just showing the kind of data we can use with EBV workflows we have on Galaxy for Ecology. So we can see that we cover different kind of EBV classes and from genetics data to more classical species-oriented data or count. And when we speak about data, we need to speak about metadata. And here we have an operationalization using the tools and services that the Data One network is proposing. So we have catalogs here for the PNDB data catalogs, where we can see, for example, that using a metadata standard internationally well-known, well-used, notably through this Data One network. So I speak about the EML, Ecological Metadata Language. We see that we have something with a standard with high power of description of data. And when I speak of data, it's data sets, but also we can also describe protocols or softwares with EML. So here I just show you what can be done using EML standards and Metacat catalog to operationalize EBV. And it's a concrete example where we see that there is a metadata file, so in EML, here in XML files, and associated data sets. So there is different kind of data sets who are EBV usable files or EBV ready files, and derived and model EBV files for visualization in PNG files. And also a workflow extracted from Galaxy, we can also describe using this amazing EML standards. So if I go a little bit further, we see that in fact, using this MetaCat solution for catalog, we can also give provenance information between tools, workflow, and datasets in front of this EML metadata standards. And we see, for example, we can describe the PNG we created with the workflow here on the interface. And here's an example of a data file is described in EML, and we see that in fact it is coming from or producing another files here, PNG files. So let's finish this demonstration or this presentation just showing that in fact often we can see EBV personalization just sharing the results and the end of the workflows described by Kissing a paper article. But in fact, here we show that we also can share and describe all EBV datasets types, from raw datasets to indicators and associated workflows. So people can reuse it and at least take it as an example, for example, to create new biodiversity observation networks. So thank you, Colin, and all the team for this work on EBV operationalization.
Thank you. Thank you. I don't think we have Colleen online, but we have Yvonne. Are you there?
Yes, I'm here.
Great. Okay, and you can hear us, I guess. So do you have any questions from the floor?
I see one question on the chat, maybe.
We have one from the floor just now.
Okay, sorry.
Oh, no, from the chat. So how do you verify the input output of the reproducible EBV workflow ? How do you cite the results?
In fact, we didn't the idea here is that the input file can be any kind of input file, and the workflow is taking this kind of input file. So input file can be, for example, Excel files or tabular files or genetic FASTQ files or things like that. And we don't have to verify it. In fact, when we created the EBV workflow, we just take a real case of EBV workflow from raw data to indicators, and then we propose this workflow to users. So if users who want to reuse it, if they don't put the good type of raw data, for sure they can't go towards all the workflows. They will have some error or something like that with message saying that, I don't know, the format is not good, or the content is not sufficient to answer the question or things like that. So quite briefly, they will not be enabled to process the workflows if the data are not conform to what is expected for this kind of workflows.
But you use things like Darwin Core, for instance, for species populations? Or...
We don't use Darwin Core as an input of our workflow because the workflow we are taking to create this EBV workflow are related to indicators created in a national scale, and in national scale, all the indicators we have are related to the use of raw data. So we are not focusing on using existing data standard, but more what is on the real life raw data to go towards production of indicators. And in the past, if there is existing workflow, if you want to reproduce creation of indicators made in the past, it's really easy because using the Galaxy platform, in fact, we capture all the provenance information inside something we call an history. So when we created, for example, each year the indicators, we capture this history and we can reuse it. There is a special button to just reanalyze all the workflow automatically.
Sorry, it's a question from Jorrit Poland, who asked the previous question. Just a clarification. My question was about reproducing the results of the workflow. that were done in the past? Apologies for my confusing question.
Yeah, so it's really, in fact, we have the workflows and we have the data, and the combination of the data flows and workflows are combined and stored in what we call an history in Galaxy. And this history is stored, and 1 year or 2 years after, if we want to reanalyze all the steps of this workflow on the data and reproduce exactly the same system, the same results, we just have to take the history and reanalyze the complete history from the same data, in fact.
Okay, thanks. Do you have any other questions from the floor? Okay. It was very impressive, Yvonne. Anything else from online? Okay, Joris going to talk to you. Okay, then perhaps we can move on to the discussion session. If I don't know if anybody wants to kick this off. Donna, you had a question more for the discussion. We could start with you, perhaps.
So I have a question regarding species identification. So currently we produce more and more data. We increase exponentially the input of molecular data based on specimens. If we can identify them, we give them names. And we know from the past that we don't really know what these names mean, because we just have a name. We don't know what is the reference, what has been used to actually identify that. So essentially it's an expert opinion, and it's a dead end in our world now, because we cannot reproduce this identification. And how can we solve that? And you've seen, we got some answers from Rod's and thing that we should actually add a reference to name. So if you actually identify a name, you should add at least a DOI for the publication, or probably better, do a taxonomic treatment. So essentially you can go back and reproduce that. Is there any thoughts given in this world now?
I hope that wasn't a question for me. Does anyone have an answer to that? I think that's a very challenging question. So I had some notes about some things we talked about that weren't related to taxonomic identification at all, but related. They are related. So one of them was bringing back Nikki's question in the earlier session about how do you give credit to people, that sort of links to what you're saying, Donna, to show the diversity of work that people do within biodiversity, and not how it all coming down to publications, but all the diverse type things we do. And it'd be kind of interesting to hear from any of the speakers today or anyone from the floor if they have ways that maybe in their organization they give credit to people for the things that are done. Does anyone have any comments on that? Particularly looking at Chuck or Joe, because they're big infrastructures.
The question of giving credit is one that we have as a question, but no answers yet. So that's, I don't have an answer, but it's something that we would like input on in the future.
I think most of you are aware of the DOI citation of data sets that GBIF does, and many of you know more about it than I do. But that's a great way that kind of is a good starting point. But there's more aspects of our infrastructures that those types of ideas, like Bionomia took one part of that and ran with it and has a great result for it. Are there different parts of our infrastructures that a similar project could be built on top to give that attribution to? I think there are opportunities there for clever people to build other things like that. And I think us as infrastructures are very interested in facilitating that as much as we can. So if you have ideas on how to build those attribution systems, for citation of scientific names in literature, can you count up how many times the species you described has been cited using the tools that we kind of have been talking about? That'd be fantastic. And within phylogenies and things like that, can we put attribution to the, the species that are being used in phylogenetic analysis as well, or the specimens and things like that. I think we're building this very slowly, but if you have ideas, don't be shy, bring them up to anybody and we can see what we can build together.
Comments?
Hi, my name is Jitendra Gaikwad, and I'm from Friedrich Schiller University, Germany. A bit of a different perspective here on data and infrastructure. So most of the time we say, okay, there is a DOI for a data set that you publish. So there you get a citation and credits. But most of the time what we do is providing as a volunteer service, and that is not something getting evaluated or credit. And there is no mechanism where people start looking at all this voluntary work as services and getting credit for the service. So like, for example, you're doing any review of let's say a data set, but then that's a service. It doesn't get counted as a scientific work, but as a real service. So we need something that can be used to evaluate that service and give credit to that service rather than scientific aspect of that data set. So I think it's just another perspective.
Hello, my name is Lyubomir Penev. So a well-known way to and widely used way to provide credit to data managers, data collectors, data scientists are the data papers. Papers that publish data, that describe data, which formally are not recognized as scientific papers in very narrow sense, but have a great contribution to science because they describe data with appropriate metadata descriptors, which means the authors of that paper get at least some credit for their really, really important and great amount of work.
We have a comment in the chat about Arctos from Teresa Mayfield-Mayer. In Arctos, they provide attribution and agent pages for work done on collection data. I think actually going back to what Joe was saying, in your data sets, you have people who publish them. And I think you can put your ORCID, for instance, on those data sets. But who is the person who goes and makes the indicators, I'll go back to indicators again, for those people? Do the people do it themselves? Do their institutions? Can you do an API that people can build a credit giving system? And where in the structure of infrastructures and the users of infrastructures are the people meant to do that? Well, to give a credit, like this person has produced this many records on GBIF, this many data publications on GBIF, etc.
I guess I'll turn this over to Chuck. So how important is that to the funders that you talk to? I mean, there's a wonderful world in which all sorts of indicators could be developed by clever people in this room. How important is that to the funders? It's important to people in this room, it's important to me, but does the NSF want funders for indicators for that type of data? Or are they happy to have indicators at kind of a data set level? Is there that demand?
I'd say that the funders at this point are much more concerned with the big picture. So this is what you're talking about is much more fine grained that they are concerned with now. If we can solve the problem of the funding and the sustainability, then maybe they'll move down to that level. But for now, their questions are, which data resources should we fund forever? Which ones are important? How can we measure how they're connected together at a much higher level?
So maybe just turning Joe's point around a little bit, maybe the question isn't so much how important it is to the funders, but how important is this attribution issue to actually solving the kind of environmental crises that we've got? Because to be perfectly honest, I don't think it is actually very central. And I know we within the community are, quite understandably very concerned about the credit problem. And there's various sort of mini solutions that we've proposed. I mean, one thing that we could do, for example, is have a better credit ontology for the collections community. So, for example, when we publish our papers, we have a credit ontology that we often use to describe how different contributions have been made to that by those authors. We could do the same for our community, but I do get a little worried that we get quite hooked up on this problem of attribution, but it's actually a bit of a distraction from really what are the bigger problems, which is actually applying or mobilizing some of this data to solve some of these big environmental challenges. And I don't think we shouldn't lose sight of that, I think, in this debate.
I guess I'm going to take the other way around. I would argue that actually it's a misconstruct that we have in the community or in our field of science, that a lot of work is done voluntarily that people do on the side, and it's a huge amount of work, and we cannot show how much work is done, and we cannot show to funders, this is how much we put in there. So I think it's essential to have these attributions very, very fine scale at the individual level. So if we have so much work to do. If you really want to scale up what we do, just developing standards or concepts or the digital extended specimen, there's so many questions that we have to solve. We need funding, and we need people to do this. We don't need machines to do that. So we need the attribution to show how much work goes in there.
Maybe. I mean, I think the business case obviously is critical, but whether that business case rests on showing the volume of contributions that we all make as a community, I'm a bit doubtful on that. It definitely contributes, no question, and it's great to have, but getting back to that NSF point, or the big national funders, are they going to kind of transform their perspective just because it shows that we've put lots of effort in? I'm not so sure. That's my personal view.
I take a compromise view. I think you're both right. Because I think it comes back to the sustainability issue. So sure, as individuals, we need credit for ourselves, and that's our own career progression. As institutions and infrastructures, we need to be able to have credit for the whole infrastructure and show we're providing value for money and impact, real positive impact on the ground. So, and both things are kind of connected. So I think you're both right. Any other comments on that? Oh, I didn't see who was first.
Thank you. I'm Joseph Fortes. I'm with IDIC Bio in the USA. And on the topic of sustainability, I feel that we should inject in that conversation words that clarify what sustainability means. From the following perspective, I think often one forgets that sustainability does not preclude renewability. Resources every 3 to 5 years essentially die on us and need to be renewed. The second thing is evolvability. The services that we provide every 5 years or so need to be completely retaught. So modernization has to be part of that definition. And lastly, as I already mentioned, it's not really, the data is extremely important, but the services that are needed to both provide the data and to use the data are really what needs to be sustained. The data is a means towards the kinds of services that we provide. So I commend the discussion on sustainability and the efforts. I just suggest that these terms be injected in the conversation.
I would like to turn around the credit thing. So it's rather who did this. So you can
scrutinize it. What's the quality? So you actually need to show, at least for data, where it came from, who did this. And that's not really the credit part, but the other way around, sort of. So it has two uses, you could say.
Yeah, with regard to sustainability, I completely agree with you. I didn't show this, but we have models of life cycles, and renewability is a good way of phrasing it. Everyone recognizes that the resources are not static. In some ways, some of the data sets are static. A nucleotide sequence data is nucleotide sequence data, but even then there's new kinds of data coming in. So yeah, we don't think that databases should be just renewed in perpetuity in a frozen state. They're always evolving, and that should be part of the evaluation. I think the sustainability issue is that we hope that the default will be that funding will continue, provided that they can show progress and reasonable development. Whereas at the moment, a lot of the funding it will stop unless you can demonstrate that you're great again every 3 years. And for an infrastructure, that's not really the right way of looking at what's happening.
Okay, thank you. I think we have some comments in the chat.
There's quite a lot of activity happening in Zoom chat. I'm going to put it into Slack if anybody wants to check it out and continue the conversation.
Okay, I think we'll wrap it up there. Thank you ever so much for coming. I have to thank Holly and Bamsey for keeping time and check on Slack and things. Thank you, my co-host Joanna as well. And let the conversation continue over coffee now. Thank you.